Hello, everyone, and welcome to our webinar for Centre UB. It's good to be back after a period of a break, after we had our coffee with fellows over the Easter and April periods. But now we're restarting the webinars, and we're joined by a fantastic talk today, which we're really excited about. Today, we're looking at research in infancy and childhood and the relationship to behavioural research. We're joined by Professor Andy Bremer, who has a career-long interest in multisensory research into this area. His career spanned work in Oxford, in London, and he joined us at the University of Birmingham in 2018. He's recently launched the Baby Lab at the university, and there's also an exciting exhibition on this at our exchange. And so today we're really interested to hear all insights about behavioural research related to infancy. So I'll hand over to Andy, and then we'll do questions and answers at the end.
Thanks very much, Vicky. It's really nice to be here and to have this chance to talk to people about my interests and what we're doing in the Baby Lab. Can everyone see my screen? Okay.
Yep.
Great, thank you. Okay, so behavioural research with human infants, challenges and frontiers. So the the remit that I want to sort of take on is tell you a little bit about how we work with babies in terms of the behaviors that they present, what kind of scientific inferences we can make about how babies sense the world around them, understand the world around them from behavior. And I'll focus in particularly on the kinds of research that we do in my particular research group on multisensory body representation. Try to clarify what I mean by that shortly. And I'm going to talk about challenges and frontiers as well. So the difficulty of doing behavioural research with babies is we can't rely on the same kinds of behaviours that we would normally rely on in adults. So often when we are trying to understand what it's like to be a human adult, we can just ask a participant to report or to respond to task instructions or to write down responses in the questionnaire, but obviously we can't do that with babies, and that's the challenge. So I'll talk to you about the ways in which we try to tackle that challenge, and I'll talk about frontiers. So in the midst of those challenges, what are the opportunities that we've got to get better and more detailed information about psychological development in the earliest moments of life? Okay. So just some definitions and concepts to start off with. So by infant behavioral research, I mean measures of observable behavior in that, and those could be in observational or experimental settings that we can use to make inferences and answer questions about how infants experience the world, their sensory, their cognitive, their emotional abilities and processes, and how those processes develop in early life. Now, this is so the research which I do really falls out of developmental psychology, or what's also known sometimes as developmental science. And also, as I'm going to be talking about, so I also kind of, as well as doing behavioral research, kind of complement behavioral methods with physiological and neuroscientific methods to look at what's going on in the infant brain. So I'll talk a bit about developmental neuroscience as well. But the fundamental questions of developmental psychologists, scientists, developmental neuroscientists, to try to understand, to study how children and infants develop. And usually the questions which are asked are try to characterize differences between babies and children of different ages. But in a way, a more interesting question, one we're always trying to address somehow, but it's probably the most challenging question to ask, is to understand how it is that development happens. So what causes developmental change between, let's say, a 4-month-old baby and an 8-month-old baby? So we know what the differences are, but how does the baby get from one place to the next? And of course, that's a complicated interaction of inherited characteristics and environmental experiences. So we're trying to understand how nature and nurture drive development from one stage to the next. I'm also going to talk a little bit about a relatively neglected question in developmental science, which is to try to understand the different ways in which different individual babies develop. We can think about that in relation to, let's say, diagnostic categories of developmental conditions and neurodiversity, but we can also think about it in terms of normative variation that we might see between individual babies across the whole population. I use the word normative kind of in inverted commas because, well, there's complications with understanding what normal is in the context of the fact that we actually vary very significantly in the ways in which we sense the world and understand it. Looking time methods are one of the key means by which we've studied infants' perception and understanding of the world. And basically those involve how long a baby looks at one stimulus versus another. And we can measure that in experimental settings and make inferences about what they perceive and what they understand of what they're looking at. And so I'm also going to, as I mentioned, talk about a couple of physiological methods for studying brain physiology, essentially, or brain electrophysiology. EEG, electroencephalography, is basically a set of electrodes placed on the scalp to measure naturally occurring electrical activity originating in the brain on the surface of the scalp. And we can use that to complement behavioral methods to understand, sometimes with a bit more sensitivity, to measure with a bit more sensitivity what's actually changing in development and how babies are responding to the world around them. OPM-MEG, optically pumped magnetometer magnetoencephalography, a bit of a mouthful, is a new brain imaging method, which I'm just going to mention briefly at the end of the talk. That's really a kind of frontier opportunity. And the exciting thing about this method is that it's rather than looking at what's naturally occurring on the scalp, it can image more directly into the brain to tell us in greater spatial and temporal resolution how nervous activity is taking place, electrical nervous activity is taking place in the brain. And I'll mention that a little bit later on. But probably the most important definition for this talk, I suppose, is to define what's an infant. So that's an important sort of starting position. So the word infant comes from Latin infans, which means without speech. So essentially, an infant is the stage of early development prior to the development of spoken language. So we know that babies recognize words, really early on in postnatal development. So they recognize words for nouns like hand and foot at about 4 months of age. So receptive language, so an ability to understand language which is spoken, has very early onset, but the production of language, of course, is something that develops gradually, kind of from around the kind of 18 sort of 12-month mark up to about 24 months of age. And typically we classify infants as being about 0 to 24 months of age, or birth to 2 years of age. Okay, so babies and infants, without speech. And the crucial thing there is we can't ask them questions. We can't ask them to report on what they're experiencing of the world around them. And that creates a really difficult research problem for developmental psychologists. So we can get around it by measuring what they sense and what they understand in different ways. But of course, then there's an added challenge because we have to then, to try to understand development, how babies are linked to what we might call a more mature state in adulthood, then we have to rely on different behavioral methods, different behavioral measures to understand how development works. What is that continuity between infants and adults? And when you change methods between infancy and adulthood, you're potentially introducing an experimental confound. Are we measuring developmental change or just different results because we're using different methods, different measures of behavioral competence at different ages? That's a complicated question that developmental psychologists kind of sort of face constantly. I think probably my view is that the best way to solve it is to make sure you're not relying on just one measure. If you rely on just one measure, then you have those kind of confounds there, and you end up with difficulties in drawing inferences. So if we try to proliferate the number of measures we have of particular sensory abilities or cognitive abilities, then we can get a more reliable picture of what it is that underlies and characterizes development in early life. So these are some of the first methods used. So in the end of the 1950s, early 1960s, Robert Fance and colleagues in the States set up this looking chamber, this infant box experiment. So they all wore lab coats back then, obviously, and it's all much more technical than we see these days. This is actually my son, my wife, Miranda, doing a more kind of modern looking time study. Obviously, lot less of sort of weird technical equipment lying around. No one's wearing lab coats, but the basis of these things is the same. What the researchers did to try to understand early sensory abilities in early life was to measure how long babies would look at particular stimuli. So if you can demonstrate that babies, for instance, prefer to look at this interesting globe-like stimulus from the bottom of this looking chamber than, I don't know, a cube that's presented on a different trial, then you can demonstrate that they're able to make a differentiation between those different stimuli. So if there's a consistent preference for the sphere over the cube, then not only can we infer that babies prefer the sphere to the cube, we can also infer that they can differentiate or perceptually distinguish between those things. Here in this particular study, this is a study looking at whether babies can differentiate different kinds of speech which are being presented to them, or different passages of speech. So you don't rely on just static stimuli, but you can also look at infants' preferences for dynamic stimuli. In this study, this is a test of visual acuity. So what the experimenter does is lift up the card, and if the baby detects this grating pattern and they have good enough visual acuity to detect that, then the experimenter should, through a pinhole in the center of the card here, see the baby's eyes moving rapidly over to the side where that grating stimulus is presented. There's been a huge amount of sort of progress using these methods since the 60s. And one of the most striking recent studies, which I was, kind of really bowled over by at the time, and really made a big impact on the field. Actually, quite a lot of people still quite skeptical about this research, but I think it's starting to be well replicated across different labs, is the demonstration that you can even see visual preferences in fetuses prior to birth. So this is using ultrasound techniques. Reed and colleagues in the University of Lancaster at the time, what they did was to shine lights into the uterus using kind of laser presentations. They would move either a face-like configuration of dots, so two eyes above a nose, versus an inverted or a non-face-like configuration of dots, so with the triangle the other way up. They would gradually move that across the mother's abdomen slowly and measure how many times the baby oriented their eyes towards the LED configuration using 4D ultrasound to scan the movements of the behavioral movements of the baby in utero. And Reed and colleagues demonstrated that fetuses at 36 weeks gestational age would make more movements of their eyes towards a face-like configuration of dots. This is what it would have appeared like inside the uterus of the baby versus that inverted face-like configuration. So this is based on the finding that very young babies can also demonstrate those preferences for faces or face-like configurations in early life. But this study extended it to demonstrate that even prior to significant visual experience, that those face-like preferences appear to be present in the infant, suggesting that there's some degree of inherited capacity to pick out faces from our visual environment, even independent of significant visual experience in early life. So in Birmingham, we have a baby lab, the Birmingham Baby Lab. We have about four quite active PIs in the Birmingham Baby Lab. Barbara Pomyczowska is my colleague who's the lead. And Barbara is interested in a range of questions about infant development, including understanding logic and understanding maths, but also understanding communicative contexts between infants and parents and infants and infants. Julia Orioli is interested in how babies perceive the space around them and how they integrate tactile, visual and tactile stimuli in the world around them. Julia and I work quite closely together because we have quite similar interests. Andrea Crott is interested in the development of language and studies in particular the effects of bilingualism on language development in very early life. My interests, as Vicky kindly said at the start, particularly in the realm of multisensory development, how babies bind their senses together. And in particular, I focused on the development of what we might call body representations. And what I mean by body representations is an ability to sense one's own body. So that's something that as adults we tend to take for granted. That this is my hand, this is my other hand, and we've got various limbs, and I can move them in certain ways in order to navigate the world around us. But of course, babies are quite motorically immature, so the ways in which they move are relatively undeveloped when infants are born into the world because we're a particularly altricial species. And I'm interested in the process by which babies learn about their own bodies and the way those bodies fit in relation to the visual world that they see and the auditory environment also. So a lot of that research involves investigating tactile perception in young babies. Yeah, just to expand on this a little bit more. So my research program kind of sort of went into this idea of studying body representations in early life in around, I guess, sort of the early 2000s or so, around 2005, I started getting interested in that. So at that point, there'd been a huge amount of behavioral and brain imaging research with young babies, 70 years or so, 60 years or so at the time, showing that infants are surprisingly competent at perceiving and differentiating aspects of their visual and auditory environments in particular. So there's been a lot of interest in visual perception and auditory speech perception in particular in early life, but very little on other senses like touch and olfaction. And the thing that I felt was really quite important is that when you're studying visual perception, baby's ability to understand what they're seeing in front of them, then you tend to be looking at their sensory development in a way which is quite divorced from their own self. So if you look at a baby looking at stimuli on a screen and they seem to understand what's going on, or they seem to be able to perceptually differentiate between particular instances of what they're looking at, then the difficulty is that we're not necessarily measuring how they understand or perceive that world in relation to themselves. They may just be perceiving that visual world in a quite divorced way from their own physical bodily reality. So I wanted to try to understand how babies develop that sense of their own bodies in the world around them. And we've done quite a lot of research on that over the years now, since sort of mid 2000s up until now. So last sort of 20 years of research on that. I'll give you a bit of a kind of a sort of flavor of some of the things that we've done. I'll talk about this study shortly, which is a touch study. I'll also talk about this study, which is another touch study looking at whether babies feel touches presented to their hands and how the brain responds to that. This is a nice example of some recent work we're doing. So a PhD student working with me, Alice Cousins, is implementing a virtual reality setup for young babies, in which what we do is present babies with images of their own body parts, in this case their feet, but we can do strange things with them. So we can present the foot bigger than it actually is, or it moves out of synchrony. So we're interested in whether the babies notice those differences between what their body looks like and what it should actually be doing, because how they make those links between what they feel with proprioception and touch and what they see. And we do that by a kind of clever set of kind of cameras and boxes and mirrors, by videoing what the feet are doing and then projecting it via a computer into a screen above, which then reflects into this mirror here. But we can distort information about the limbs. I won't mention this study for the moment, just in the interests of time. So this is one of the first studies we did with 6-month-old and 10-month-old babies. What we were interested in was whether babies could localize touches on their hands. Hadn't really been asked very much before in 2008. So hopefully, I'll sort of wind that back. So, so when you see the word buzz up here, that means there's a little tickle going off in this hand, a vibrotactile stimulus in the hand. Hopefully you can see that baby notices that really quite clearly, that there's a very obvious manual response to that. What's also interesting though is, so if you or I were faced with that situation, an unexpected tactile stimulus in one of your hands, an unexpected touch in one of your hands, you'd probably immediately look towards where you felt it. I think that's probably what I would do. But there's not much of that kind of behavior here. So if you watch the eyes, you don't see very much visual orienting towards the touch at all. So the hand moves, and then eventually the eyes start to move towards where the hand's moving. And that was quite characteristic of all of the 6-month-olds that we tested in that particular group. So if they felt a touch in that hand, they would respond quite strongly with the hand, but wouldn't move their eyes towards it. When they did move their eyes towards it, it tended to be after there'd been a long period of flapping with the hand, almost as if the movements of their hands what attracted their visual attention towards it. But in contrast, 10-month-old babies showed a very different behavioural pattern. If you present a touch in their hand, 10-month-olds would immediately look with their eyes towards where they felt that touch happening. And it was all, yeah, so the look would actually happen in advance of the actual movements of the hands in the 10-month-old group. So what we seem to be measuring there was quite significant developmental change in a visual awareness of the body between about 6 months and 10 months of age. The young babies appeared to be aware of the touch in terms of their body's responses, but not able to locate that touch very, very quickly or very well in vision. But the older babies were very rapidly using their vision to locate a touch which they felt on their body. So perhaps what we're seeing is a developmental integration of the visual world with the tactile world of the body across that first year of life. And that delayed development suggests there's probably quite an important role for multisensory experience in teaching babies about their own bodies. So we followed that particular study up in a range of ways. One is to really kind of complicate things. Well, this is on the feet this time with 4-month-old and 6-month-old babies. One thing you can do, which confuses adults actually, you cross the hands over and then we start making mistakes about where we felt a touch. That's because when we feel a touch in this hand, it usually happens over here. If we feel a touch in this hand, it usually happens over here. But if you obviously cross the hands over, then that relationship between your visual environment, your spatial visual environment and your body is in conflict with the usual state of affairs. So it's a bad idea to try riding your bike with your hands crossed. Don't try it because you fall into those kinds of mistakes and end up crashing to the ground. But it seems that that's something that we learn. So we have to learn about the relationship between our tactile body and the visual environment. So what you might predict then is that young babies wouldn't show this crossed hands effect. So they wouldn't be confused when their hands or their feet are crossed. And that's in fact what we did find. 4-month-old babies, whether their feet were crossed or uncrossed, were just as accurate at responding to a touch on their foot. Whereas 6-month-old babies, if their feet were uncrossed, were just as accurate as the younger babies, but when their feet were crossed over, then they were at chance. So if their feet were crossed like this and they were touched in this hand, they were just as likely to respond with this foot as with this foot. Sorry, I'm mixing up hands and feet because we've done too many of these kinds of studies. More recently, we've followed that up to investigate what the role, what it is that's leading to these developmental changes between 4 and 6 months of age, where you learn about the relationship between the body and the world. And that's been work carried out with Monica Gori and colleagues in the Italian Institute of Technology in Geneva. And Monica works with blind children and infants, and is interested in the role that visual experience plays in the ways in which we link up our senses together, or in the ways in which we learn to respond to senses. So Monica has studied this same manipulation, this time with hands rather than feet. So we're interested in whether babies will respond to a touch in an uncrossed hands posture versus a crossed hands posture. And they replicate this finding with older babies that if you're a sighted baby, so the purple bars here, you're much more accurate at responding to a touch when your hands are uncrossed than if they're crossed. But if you have a visual impairment as a baby, so a peripheral impairment of vision, so you don't get visual experience in early life, then babies show enhanced performance compared to the sighted babies in the crossed hands posture. So they're better able to locate touches when their hands are crossed over. What that indicates is that visual experience is really important in teaching you about where the hands are in left and right space. And it can actually lead, in the case of vision, to declines in performance when our hands are in unfamiliar postures. Whereas if you have visual experience, then whereas if you have visual impairment, then that visual experience doesn't teach you in the same way to expect touches in this body part to be over this side of visual space. What this indicates, I think, for me I think it's kind of sort of fascinating insight into the differences by which we can sense the world around us. We can have very different qualitative representations of the relationship between our body and the external world, depending on the kinds of sensory experience that we've grown up in. Okay, and I'll just move on from this one. So how am I doing for time, Vicky? I don't want to run over time too much.
You're okay, about 5-10 minutes.
Great. I think I'll probably be another 5-10. Yeah, I think that'll be about right. Yeah. Okay, so I wanted to tell you a little bit about physiological methods. So you might think, well, so let's talk about behavior. This is the Center for Understanding Behavior. So why are we talking about physiological or brain imaging methods? And I guess my sort of coming back to one of the things which I was saying early on in the talk about the importance of getting lots of information about how development happens, because we have relatively little to go on in babies because we can't ask them questions. So we need to get more information from lots of different kinds of measures. So I'm a strong believer. I don't think this is always the case through my career. I was a bit skeptical about what could be added by physiological methods, but I'm more and more persuaded that by adding physiological methods to our behavioural methods that we can use for measuring development in early life, then we get a much richer picture and we are much better able to interpret the behavioural findings that we get. So we've used EEG to try to understand changes in the ways in which touch is processed in the brain in early life. Now, the somatosensory system kind of runs across the top of the head here. So you have somatosensory cortex running down what's called the sort of central sulcus. And what we can do from that is to place an electrode cap on the head and get that electrode cap to measure a somatosensory evoked response every time the infant feels a touch. So what we should see is what you can see here. When you present a touch is a kind of hot spot of activity over the central part of the brain. So this is in response to a tactile stimulus that happens here. We're seeing a hot spot of activity over a particular part of the brain. So that's contralateral. So this is when a baby feels a touch on their right hand. It crosses over the brain to appear in contralateral somatosensory cortex. So what we can do then is look at the pattern of activity in time. So at this point here, this is when a touch is presented to the hand, and we can look at what are called somatosensory evoked potentials. So it's a kind of wave of voltage changes measured over these central regions in response to that somatosensory stimulus. And what we did here was to compare 6-month-old babies and 10-month-old babies. The 6-month-old babies, and for both groups of babies, we compared their brain responses to touch in a crossed hands posture and their brain response to touch in an uncrossed hands posture. The interesting thing here is that the 6-month-old babies' somatosensory evoked responses are exactly the same according to posture. Oops, sorry. So it's as if the brains don't care whatsoever that their hands are in different postures. They process the touch in exactly the same way. But the 10-month-old babies, on the other hand, show a very early modulation in the way their somatosensory cortex processes touch according to the posture of their limbs. And what we've concluded from this, based on what you can also observe in adults, is that an early is a sensory mechanism for processing the current posture of the limbs affects or modulates the way in which that touch is processed. It's as if the brains are trying to take into account the fact that the touch is occurring not in its usual place, but in a different place in space at around 10 months of age, but not at 6 months of age. But the thing that's really interesting for me is that difference in the way the brain processes across those two different posture conditions is really early on in the somatosensory processing stream. So if we observed this difference, but it was really late on, so it's towards the end of sensory motor processing, then we might conclude, well, what we're seeing developmentally between 6 and 10 months of age is a change in how babies are behaviorally responding to those somatosensory stimuli, not to how they're sensorially perceiving those somatosensory stimuli. But the fact that we can see this developmental change occurring really early on in the brain's processing of the stimulus indicates that what we're seeing developmentally between 6 and 10 months of age is a change in the way babies are sensing the somatosensory stimuli that are presented to their bodies. So that's one of the problems with behavioral methods with babies is that what we're always measuring is the end point of then sensory followed by cognitive followed by sensory motor processing. And so any change that we see behaviorally through development could be the result of any of those aspects of mental processing. But the beauty of EEG is that we can isolate which part of which part of brain processing of sensory stimuli is changing with development. And in this particular study, it indicates that it's sensory changes which are going on. So to draw to a close, I want to talk a bit about some of the frontiers that we have in developmental psychology. One of those is trying to understand. So, a lot of what people have done up until this point, particularly in the world of sensory development, is to understand what happens usually at 4 months of age, what happens usually at 6 months of age, what happens usually at 10 months of age. And the studies which I've just been demonstrating are no exception to that. We're demonstrating what we might call a normative picture of development. But of course, babies aren't all the same. Babies develop differently. And it's increasingly apparent that in my particular area of interest in sensory processing, that early sensory processing differences could lead to really different outcomes in terms of behavior, but also in terms of mental health, and in terms of neurodivergency also. So adults increasingly report, adults with neurodevelopmental conditions increasingly report that sensory processing differences are some of the key difficulties that they occasionally face. So I think a really exciting potential there is to try to get past that normative study of developmental change to understand how infants differ at a given age group and how those differences remain stable or vary across different periods of development. And I'm at the moment in the process of proposing and hopefully gaining funding for a set of studies to try to answer these questions about how individual differences in sensory processing are present in the earliest moments of life and how they change. One of the ways in which we're hoping to do that, and this is work which is really led by my colleagues at the University of Birmingham, Barbara Bomichowska and Anya Kowalczyk. It's a collaboration between neuroscientists, developmental psychologists, and physicists, is to use this new method of optically pumped magnetometer, MEG. So the good thing about MEG, it measures it measures brain activity through magnetic fields. What's going on here is a laser is shone into one of these sensors which are placed over the infant's head. And there is some alkali metal vapor inside one of these sensors. And those alkali metal atoms are disturbed by magnetic fields arising from the electrical activity that's going on in the brain. And those magnetic fields are not disturbed by passing out through the skull, so we're able to get a very detailed picture of what's going on in which part of the brain, much more detail than we get from EEG, which tells us really just what's happening when, not particularly what's happening where. So OPM MEG is also highly sensitive, much more sensitive than other forms of MEG or even fMRI at detecting particular patterns of neural activity. And the good thing about it in comparison to things like MRI or more traditional MEG is that you can place the sensors directly on the baby's head in a relatively flexible headgear. MRI, as you may be aware, when you're having a head scan in MRI, it makes a very loud banging noise. You're in a very kind of enclosed MRI environment. Babies don't like that very much unless they're asleep or sedated. So MRI has been really difficult to kind of get going with young infants. So OPM MEG is really exciting in that regard because it's a very sensitive method. So that sensitivity gives us something which enables us to be able to prize apart with some high power differences between individual babies in terms of the ways in which they're processing their sensory worlds. So that's very exciting for me, and I'm very lucky to be kind of associated with this excellent collaboration between developmental neuroscience and physics in trying to create these new infant scanning setups. That's really just a picture of the baby in the OPM setup in the University of Birmingham, and a demonstration of some of the data that we've gathered already from young infants, but I won't go into the details for that for the moment. It's quite early days still. And yeah, so I was asked just to give a bit of a sense of, what are the impact what's the impact relevance of this research? What are the potential sort of policy outcomes? And that's something that I've tended to focus on what we might call fundamental or basic research over my career so far, but I'm trying to make more and more of a step into thinking about how the work that we've done in asking these fundamental questions about body representations, about sensory development, can go on to actually influence policy or to be helpful and useful in the world. And as I mentioned before, there's this increasing recognition that sensory processing differences in early life have quite important developmental outcomes in terms of neurodivergent outcomes, in terms of mental health outcomes. And I'm working with a range of people, colleagues at the University of Birmingham, like Andy Surtees and Caroline Richards, to ask a range of questions about how sensory processing in early life is related to neurodiversity, to mental health outcomes. We have one PhD student, Andy Surtees, and myself working in the mother and baby service at the hospital just across the way, to try to understand how neurodiversity in that context could lead to different kinds of sensory interactions between parents and infants. And with Andy again, and with Alice Jones Bartoli, funded kindly by the Centre UB doctoral training partnership, doctoral training centre, we're looking at the role of sensory processing differences in young babies in informing to try to understand the interactions between infants and parents as they get older. One of the policy implications there that we're exploring is that, I mean, you may well be aware, especially if you've had your own children, that you often get advice on how to bring up children in quite a one-size-fits-all kind of formulation. But of course, given that infants differ, given that parents differ, sometimes according to neurodiversity, then perhaps we need to be thinking about different kinds of advice on how to get babies to sleep, different kinds of advice on how to interact with children and so I'm working with Andy to try to get some nice data on individual differences in infant-child interaction, and we're working with Alice Jones Bartoli at the National Children's Bureau to think about how we can develop policy implications for that kind of research. Great. And I think I'll stop there and move on to the question and answer session. Thanks very much for listening.
Brilliant.